I'll go second because my name starts with B. I'm Brian. I'm a scientist at Agilent Technologies, the same company that Robby virus works at, and we're friends. Anna's actually my partner. So anyway, I got a PhD in biochemistry a long time ago, and broadly I study measurement of molecules. That's kind of my job. It's like we have these different molecules, these biomarkers, like what are they? Mostly I specialize in DNA. I spend a lot of my day job trying to work on things that may lead to better cancer diagnostics. That's kind of what motivates me. But last year, COVID, all right, yeah, I mean, the virus is RNA, but tests are tests. It's nucleic acid. And so, Rob and I and some other people at our company tried to help our company navigate, like, we sell instruments, we sell tests. How are we going to deal with the pandemic? What should we do? Currently, one of my fractions of science time is to think about, I'm working on a T cell test, which is a little jargony, but we are trying to test not just like, do you have the virus or do you have antibodies against the virus, but are the living parts of your immune system still, like, how active are they at working on the virus? And it's kind of a complex test. That's why they're not really commercially available yet. It's a lot of researchy thing. But anyway, I'm along for the ride. Plus, I made drinks and coconuts. So.
Very cool. Thank you. Doug, you're up.
I actually really love geeky things like that. And a T-cell test would actually be really, really helpful right now. Of course, then we'd have to really explain what T cells do and what's the difference between T cells and B cells. And we still can't get people to keep straight the difference between antigen and molecular and antibodies and infection and all that fun stuff. But that's why we're here. My name is Doug Levy. I was a reporter for a long time, most of that time covering the medical research world and the Food and Drug Administration when I was at USA Today. And then I went on to work I actually worked at Johns Hopkins and later at UCSF and at Columbia University Medical Center. I kind of spent about a dozen years in the weeds of medical research, although, I'm in awe of the people that do the actual science. I help communicate about the science. I'm a wordsmith. I'm intensely curious, and I like helping make things that are complicated a little bit easier for people to understand, especially when it comes to healthcare, because I believe that better information gives people the power to make better decisions to live healthier lives. That's kind of the reason I got into journalism. I also kind of got into pandemics because my job at UCSF included emergency management. And when H1N1 came about, I found myself on that little committee that had to plan for what if it got really bad. And then in New York, one of the physicians in our university got Ebola. So we had already done a lot of Ebola plans and prep, and we wound up in the middle of it. So here we are. These days, actually, I do need to do some quick disclaimers. I am a lawyer trained in healthcare law, but I am not practicing. I am not here as a lawyer. So anything I say, it's not legal advice. And I'm also not a doctor. So I'm going to tell you what I think and know based on my reporting and the work that I do with scientists who are involved in COVID work. But for God's sakes, for your own health decisions, consult your own healthcare professionals, not Dr. Google, please. Anyway, I'm happy to be here. We'll have a great discussion, I think.
Awesome. Yeah, it's so great to have you join us, Doug, and Anna officially join us. Because last time you contributed, but you weren't an official panelist. So it's great to have both of you.
Yeah.
Cool. Robby virus.
That would be me. First of all, I have to disagree with what Doug just said. I think you should get all your medical advice from Facebook and or Twitter. Maybe Carlo if you kind of swing that way, but not on people like Brian and me. I'll just lead you down the rabbit hole.
Well, actually, so you're making a really, really good point. There are some really credible experts who are accessible on social media.
That's true.
But there's also fakers.
Yeah, and if you don't,
Mhm.
if you don't know
Yeah.
the field, you probably can't tell the difference. That's the
Just because somebody has an MD or claims they're a doctor or something, they might even be an actual MD. They still might be full of garbage.
very true.
Mhm.
Okay, I'm off my soapbox now. Go ahead.
Yeah, so I work with Brian at Agilent Technologies. I'm a molecular biologist. Did my PhD at Yale studying RNA production in mammalian cells. Then came out to Berkeley for a postdoc and switched to plants, studied genes that control the cell cycle and growth regulation in tomatoes. And then switched back to mammals and human genetics when I came to Agilent. And yeah, I do a bunch of stuff, work on measuring, how to measure DNA, how to look in tissues and see what genes are being turned on and off in a particular tissue. And yeah, since, COVID started, I mean, pretty much everybody in the field is like learning about COVID. So that's
Perfect. Thank you so much. So there were a bunch of questions that were in the.
and I'm drinking a margarita.
Oh, right. You're drinking margarita. Doug, you talked about what you're drinking earlier, but you didn't elaborate what you're drinking now.
I'm drinking an organic turmeric lemonade. It's actually very good. It's very exciting. My day starts at 5 a.m. because I work with folks mostly in Washington these days. So I don't get to drink on most days, most weekdays at least. I'm jealous.
Well, maybe if we do another one, we'll have to do it on a weekend night so that you can kick some back. Well, I wondered if the panelists want to talk about where do you believe we should begin tonight? We do have questions that are in the Facebook room, so we could start with those questions. Or Anna, you could start with your PowerPoint and talk about that if you'd like, the slides that you created. But since you guys are at the forefront of this, what are the issues that you're seeing now that you think are relevant to start off the discussion?
So as a part of the test that I created, I do a weekly analysis of the sort of circulating virus and the genomes that we're seeing in the circulating virus. So obviously, Delta is a really big proportion of that. One of the questions was in the chat was, what percentage of new cases in the US are Delta? And I have a slide about that. I think, so it's interesting because the virus didn't really evolve that much at the beginning. There weren't that many changes. But now I think there's two things. One, we're seeing people who have had a COVID infection for a long time, like immunocompromised patients. So it has the opportunity to sort of learn a little bit better. And I think also now the virus is being challenged. When this first started, everybody was susceptible. So the virus didn't really need to change because it was already good at doing what it needed to do. And as it's starting to face more challenges, as more people have had the disease or been vaccinated, that's when we start to see these collections of variants that are better at infecting, better at spreading. And I think it's going to be really hard to get control of the situation unless we get more people vaccinated, because otherwise it's just going to sort of have more opportunity to learn to get around that. And we don't want that.
Yeah. I mean, I just read an article in The Atlantic today that reiterated or sort of is next to some of the pieces that you're talking about vaccinated versus unvaccinated. Do vaccinated people really, are they as contagious I don't know if I don't remember the date. I don't think I looked at the date of when it came out, but I know that that's certainly an issue. I'm so curious about how you can tell that something is Delta. They're actually doing the genome tests of that now?
Yeah. So
Yep.
I will, I'll talk about that a little bit in my slides. So maybe I should just go ahead and pull up my slides. Does that sound good?
Absolutely, go for it.
Sure.
Awesome. Let's see. This is a Windows machine. I always have to remember how to do that.
Go to.
Oh, no.
Did you
no,
save it?
Let me.
Okay.
Share screen.
Should be able to.
Oh, I clicked that. Whoa. Oh, no. There we go. Share.
That one.
Ooh. Oh, where is the, the correct click? Yeah, I think that makes it big enough.
Yep.
So I tried to answer a lot of the questions that are in the chat, and the other panelists can feel free to jump in. I've highlighted a few points where like Rob or Brian might know more than me, especially when it comes to like neutralization tests, because I work with genomes and not immune cells. So I will post this in the chat. These are some resources that I find really useful. Obviously, there's a lot of resources, but these are resources that I trust. So one of the questions was about like, can the vaccine cause problems with your DNA? Like, can it cause birth defects and things like that? So I like to think of the cell as being, the cell is sort of a house, and the nucleus, which is where the DNA is sort of like a safe room in that house. Things come out of it, but it's really hard to get into it. So mRNA really only goes one direction, which is out of the nucleus. When you get vaccinated, you can see sort of the outside of the cell is where the cytoplasm is. So that's where the mRNA, the vaccine, gets into. But because the nucleus is sort of a safe room, the mRNA can't get in there. So it doesn't have the opportunity to change your DNA. It's like it's completely locked out from the library, so it can't do anything. It can't alter the books or anything. So, you know, all of the science suggests that there's no reason it should cause things like birth defects, at least from sort of a genetic level. I don't know if Doug or Rob or Brian know anything else about, like, I think in terms of antibodies, we've just seen like getting the vaccine when you're pregnant puts antibodies in your breast milk and in your baby, which is good.
I mean, that's about that. That certainly is accurate. And the thing is that you know, the messenger RNA is just that. It is a messenger. It is delivering information. It is not itself doing anything inside the body. It's merely putting the information there as if the virus was there, except it's not the virus. So it's giving the information that the immune system needs in order to create the reaction to fight the virus.
So in that way, is it different than traditional? It's different than traditional vaccines, which are, from what I understand, they're non-active virus. This is not even that.
That's not. So neither of those is actually right.
Okay, good.
Actually,
yeah.
if there's one of the scientists is probably better at explaining vaccines than I am, but
I mean, there's a lot of different types of vaccine. One of them is virus that is inactive or killed or weakened in some way. And so that is, you know, put up your nose or injected in your arm, that gets into your cells, and your cells are like, oh crap, virus, and make a response. But because it's weak or killed or in some other way doesn't work, you don't actually end up getting sick. You might have, like, you know, sometimes when people get the flu shot, they get like a headache because your body is having an immune response. It's just that like the virus can't make more of itself to continue to infect you. So that's different because it's sort of like putting the whole like car into your body versus like just putting the manual for the car.
Right.
And,
Yeah.
and
I guess
the Johnson, go ahead.
I was just going to say there's lots of different kinds of vaccines. I think one of the things about this shot, like the COVID vaccine, a lot of people maybe aren't aware of how many vaccines. It kind of depends on your age and what country and what part of the world you're from. But if you're as, you know, if you're 50 years old or younger and you were born in the United States, you got a whole bunch of jabs when you were a little baby. You probably don't have your vaccine card, but I have my son's vaccine card, and I counted 19 different things that he got vaccinated for. And I started to break this down. It's like, how many of them were weakened to virus? How many of them were killed by it? And it's complicated, but there's sort of like a mix. Sometimes they just use a protein. Sometimes they use a dead virus. Sometimes they use like a weakened virus. Sometimes the flu vaccine, they're like, oh, do you want it up your nose? Sometimes that's weakened virus. Or do you want it in your arm? Oh, well, that one's dead.
Yeah. And
the mRNA is a little bit different, but not really. It's sort of like, oh, in this rainbow, we have a new color of blue. It's not that different. It's kind of like.
What is significant about the mRNA is the manufacturing. So for the adenovirus vaccines, like the Johnson & Johnson and AstraZeneca vaccines, and several of the other ones that are in development, there's five major vaccines in development in the United States right now. We've got 3 that are authorized. 2 of them are mRNA vaccines, the Moderna vaccine and the Pfizer vaccine. Both are mRNA, which is a technology that's been in development and some use, well, in development since about 20 years ago. And its first use was, I believe, there were several Zika vaccines that went into clinical study. I don't know if any of them, I'm not sure how far those got, but the clinical research on the Zika mRNA vaccines made it possible to pivot to a COVID vaccine very quickly. We can talk more about that if anybody wants. The Johnson & Johnson shot and the AstraZeneca shot both use an adenovirus, which is basically a very weak cold virus that can't get you sick. That takes longer to manufacture because you have to grow those cells before you can get the vaccine distributed. mRNA can just be manufactured. So you can literally go from having the drug, figuring out what needs to go into it and making it within 30, 40 days, I think, versus 70 or more just to do the cell cultures for the other vaccines.
Yeah, it's
that's
really
why the
cool.
Johnson & Johnson shots are behind.
Yeah, there's a really cool, I think it's Pfizer did this photo essay with the New York Times of like what goes into making the mRNA vaccines. And it's still a lot of steps and tubes and liquids and dry ice, but it's a lot faster.
Yeah.
Got it.
Which is also good news for, when we have things like variants and other diseases, because, we can just print a new one and try that.
Well, and that's also why the folks that do pandemic work have really been looking at the mRNA platform as one of the things that could really make a difference in a pandemic. I mean, the federal government's been working with Moderna since 2015, I think. And that's how this came about. They were working on Zika, they were working on Ebola. Now COVID.
Your tax dollars at work.
Yes.
All right.
Robby,
Do we have another question?
Robby, you've been working with mRNA for 20 years, right?
Yep, that's true. Yeah, I just want to add, when you're infected by a virus, a cold virus or gastrointestinal virus that causes diarrhea or whatever, that virus gets in you and it makes its own mRNA. So, this is a natural process and you're exposed to these foreign RNAs all the time. And, they're not, when you get a cold, that cold is not genetically changing your DNA. So this idea that mRNA changes your DNA, it just doesn't happen.
There's a lot of science fiction out there.
Yeah.
So I wanted to talk a little bit about detecting COVID versus, really loud, versus classifying a variant. So what usually happens when people are detecting if you have COVID, you go to the doctor, they do a swab, and then they do something called a nucleic acid amplification test, or NAAT. That's usually what you'll see on your doctor's report. Most commonly, this is a process called qPCR. So you take a few small fragments of nucleic acid from, so you take the virus, you purify the RNA, you make it into DNA because it's a lot easier to amplify DNA than RNA. And then you amplify little tiny portions of it, usually about 100 nucleotides, so 100 letters long. And as it amplifies, that actually causes fluorescence or light, it glows. And so they can basically say like, oh, you know, we did 10 rounds of this. And at that point, your sample was bright, glowy enough that we can say that you have virus. And you can actually back calculate to say, well, that was this many amplifications. So you had this many copies of the viral genome in your sample. It doesn't correlate exactly to like number of virus because it's complicated. But it tells you if you have the virus, but it doesn't tell you what variant you have of the virus. Because it's only looking at a couple hundred bases and its readout is glowing, not what we call sequence or the sequence of letters of the genome. So how do we tell variants apart? There's a lot of text on the slide, but that's because I really want to be thorough. So the SARS CoV2 genome is 30,000 base pairs or letters long, so the ATCG of DNA. There are between 17 and 36 differences between the first SARS CoV2, so what we first detected in Wuhan, and Delta. So one option is you can do the glowy method, but when you try and amplify it, put the mutation under the little piece that binds for amplification. And if the mutation can bind, we know the mutation and glows, we know it's there. If it can't bind, then the mutation isn't there, it doesn't glow, so you can tell them apart. It's sort of like saying like, well, I have a drawer of 10 socks. This sock is long, this sock is short. And since I have these combination of socks, I must have bought socks from this store. It is very fast. It uses the same test as the detection. So a lot of people have the equipment already. But do you do it for all 28 mutations? What about new mutations? It's not a super good approach. Another option is if there's only 28 changes, well, just try and read those exact 28 changes. So there's this older method called capillary electrophoresis DNA sequencing. It can read chunks of DNA, sort of 500 letters at a time. And that's really good. If you've got, you know, lots of mutations next to each other, you can capture them all at once. So it's good. You don't have to do 28 reactions. But again, it's only the mutations that you know about, right? That's a problem. What about new mutations? A lot of the stuff that came up in Delta, you know, we never knew was going to be there. Also, a lot of facilities don't have these machines, and it's kind of slow. So I'm biased because this last one is the one that I worked on. I feel like I have to disclose that. So your third option is to try and read the whole genome, just the whole thing all at once. You can do this a couple of ways. You can break it into pieces and read those small pieces. You can amplify small pieces of it and stitch it together. You can use, there are special machines that can read a lot of letters all at the same time. It's sort of like when they expanded the text limit for Twitter from like 160 to, I think it's like 320 now. You know, it's the better Twitter. So this is great because you detect all of the mutations and all of the new mutations, right? All the stuff you would never know about. it is more expensive and it is slower. A lot of hospitals don't have these. This is also the kind of testing that they often do for like cancer. So, you know, your hospital in Riverside, California probably doesn't have a machine to read all of the details, so it has to be sent somewhere else. But this is actually considered the most useful of any of these methods. So several months ago, the CDC pledged $200 million for this method. There is one particular company, Mako, that does this. They have increased from doing a couple hundred genomes per week to doing about 10,000 genomes per week. So huge increase. They've got 3 shifts of people working through the night to try and sequence as many of these things that happen, because there's hundreds of thousands of cases in the US, but we only sequence a portion of them.
And that's only been since January, really, because prior to January, I don't know what happened in January, but somehow we went from ignoring all the reality to actually monitoring and testing throughout the country to find things like variants.
Yeah, so I'll tell you 2 things that happened. One, a variant cropped up, beta, that started causing some of the detection tests to fail a portion of the time. And they were like, oh my God, this is a problem. We need to like check all of these ones that fail and see what they are. Also, there was an opinion piece in one of the leading journals that was like, you know, it's weird. The UK has a really small percentage of like the people who live in the world, but they've sequenced more than like 50% of the COVID genomes that are out there. I wonder why that is. Why is the US so behind?
Anybody want to guess?
Yeah. So, you know, some things happened, but it's exciting that now, you know, we're starting to like pick up on this and get much better at it, because I think it's going to be really important to understanding exactly what's out there and how to combat it, right? If you don't know what the enemy is, how are you going to fight it? Any comments from?
Testing has been a bit of a problem for the United States.
Yeah, it's interesting because like even a couple of days ago when I was at the doctor, like they've got all of the supplies for the rapid test, except for the nose swab. We've run out of nose swabs again. So we still got them for the like qPCR, just not the rapid one. So
different swabs.
Yeah, Q-tip, because I think that's the brand name, has actually transitioned a couple of their plants from making Q-tips to making medical swabs, because Q-tips are cotton and medical swabs are some sort of plastic, and that makes a big difference in how you transport the virus.
Yeah, they can't be reactive.
Yes, exactly.
And they also have to be individually wrapped, which is a problem. Sorry, I'll stop interrupting.
I actually really enjoy, like, I think it's really smart the way that they've designed those packages that you can just like pop them open with just like one hand. When I worked in a lab that dealt with a lot of human blood, you really come to appreciate the stuff you can do one-handed, because you're like, oh God, there's blood on my hand. I need the other hand.
One of the positive things, if I can say that, that's going to be coming, or that is already coming out of COVID is, you know, there are things being figured out now that probably should have been figured out before, but okay, we're at least figuring it out now. Like the low dead space syringes or vials. Nobody even knew what that, nobody had even heard of that before. Is that even a thing? And now we've got factories making vials for drug product that are more efficient. That's going to help.
I think it also helps that we have more awareness of sort of epidemic, pandemic testing vaccines, antibodies. I mean, these are important things. Generally, they're kind of, at least in the United States, they're kind of going on in the background. You're like, oh, there was something about Ebola. Yeah, that sounds nasty. I don't know. Hope they fix that. Are they going to fix that? Well, vaccines, but it's hard, but it's going to happen again. And there's drama, there's miscommunication. In 1918, there was the flu. It wasn't that. It was like, oh, all the citizens listened to whatever the government's not that well-informed advice was that followed it. There's all kinds of theories. It's just, it's the same thing happening over and over again. But
I'm gonna see if I can share this in the chat real quick. So then people can always I don't know how much of it we'll get through, and I don't want to bogart the
yeah, you sent the link. So also questions from the people who took the time call in and listen.
Chat. Oh, there we go.
Are there any live questions?
Yeah, so folks, I know that some of you have put things up in the Facebook invite, and I can pull some of those up now. Robby, you answered one of them. I don't remember what it is offhand, but I know that some of you who are on here now have asked questions either in that or they've asked questions at other times. So,
I see a question in the chat, which I can answer. The difference between the Pfizer and Moderna vaccines, and why are we hearing all about Pfizer and not about Moderna? It's a question of timing of data and when clinical trials began. Pfizer got their vaccine into clinical study about 3 weeks faster than Moderna. And as a result, they got their results about 3 weeks faster and got their emergency use authorization faster. Moderna has submitted data to the FDA for boosters and for full approval. The FDA is considering those things. We're going to be hearing a lot about that within the next couple of weeks, and J&J will be right behind them. So it's just kind of the bureaucratic and scientific process going in sequence, which makes it really confusing for everybody. But it's not a question of one vaccine necessarily being better than another. There haven't been any side-by-side head-to-head comparisons. So it's really hard to say that one is better than the other. Although we are seeing some data that at least suggests the immune response is more durable, lasts a bit longer at a high level for the J&J shot and the Moderna shot versus the Pfizer shot. But something that I think is getting lost in a lot of the translation is all three of the vaccines, including Pfizer, even if it does wane somewhat after 6 to 8 months, it still is giving a very high level of protection. And the reason the CDC is not authorizing boosters for everybody, or even a lot of people, is that there's not enough data to show that the benefit of those boosters is all that much. You're still in pretty good shape if you've been fully vaccinated with any of the 3 vaccines that have been authorized in the US.
Yeah, I'd agree with that. And yeah, we're seeing, I mean, you hear about breakthrough infections but and people are getting these, and vaccinated people are getting infected, but like the number of breakthrough infections that result in hospitalization and worse than that, death, like still very low.
It's not zero, but it is very low.
Stepped up a little, but you're still well protected. And yeah, I agree. I think the data on boosters, like, do we need boosters? The boosters, how effective are boosters? We just don't know yet. I mean,
one of the interesting things at the FDA hearing last week, where among the things that was presented was the Israeli data. Israel has had a very powerful vaccine program. They got a lot of people vaccinated very quickly, and they observed some waning immunity and started a booster shot program, and saw an improvement in results, in fewer people getting sick and dying. But the FDA experts, evaluating the data, once they were able to really go into the detail, we're able to see, wait a minute, this is not applicable to the United States because the way our vaccine program has rolled out and the way our population has responded to COVID is different. So the fact that we have communities that are not taking precautions, not wearing masks, not doing social distancing, not getting vaccinated, means that the risks here are very different from a place like Israel, where you've got very high percentage vaccinated. So here we've got a much bigger risk from new variants than anything else. And we've got to get those first shots into people's arms.
Yeah. So Doug, I have a question along those lines. Have you heard anything? So Israel's giving people that have had the 2 shots now a booster. Have they had any cases of people who got the booster but then got breakthrough infections?
Yeah, I'm going to have to check my notes from last week. I don't think so. They had a very small number of adverse events. You know what? I'm going to see if I can find that answer. So if you see me looking to a different screen, I've got multiple computer screens here. I'll see if I can find the data that the Israeli health officials presented last week and look that up, because I don't remember it being a significant part of the conversation, but let's find out. Good question.
So yeah, a question that I have, which I don't think anybody's answered yet, is, when if you give people 2 shots and then you give them the booster, like, are they then really immune and you're not going to need another booster for a long time? Or is it, we're going to need the boost every year?
I can do that one. Actually, is there an immunology person who wants to jump at that? Because I don't want to monopolize, but you're asking me easy questions.
You go for it.
Okay.
I'll cross-check you. I just took Immunology 101, but I only got halfway through the online course. But anyway, fresh.
You probably did better than I did. There are a lot of vaccines, like the hepatitis B vaccine, which I remember I had to get when I was an EMT. That's a 3-shot vaccine. You get, and I think it still is, you get dose 1, then dose 2 about 4 weeks later, and then you get another shot 6 months later. It's very likely that that for COVID, we're going to need something similar. What happens with the COVID vaccines is that the first shot gives you, kind of wakes up the immune system. And from what I've seen, the data suggests that first shot gives you an immune response kind of along the same lines as if you had had a natural infection with COVID. So it's an antibody response, but it's not enough to hold off another infection. The second shot, 3 weeks or 4 weeks later, of the Pfizer or Moderna vaccines, amplifies that response. So it's like the immune system is already listening or watching out for that spike protein. And then when it gets that next dose, it's ready to jump. And you wind up with 5 or 6 times more immune response. And that's a level of response that protects you from infection, and including against even the Delta variant. The data that we're seeing suggests that the third dose, 6 to 8 months later, at least brings the response back up to that high level after the 14 days after the second shot. But Johnson & Johnson shared some data yesterday in a press release, so we haven't seen the underlying data. But the Johnson & Johnson news release said that their booster shot actually multiplied the immune response by 12 times, which would be pretty good. Now, other vaccines with that 3-shot regimen tend to be very durable. So you get the 3-dose series over the course of 8 months, and you're done for years. And that may very well be the case here, unless there's a lot of variants, in which case we'll be getting shots every year probably.
Yeah. And I think both Pfizer and Novavax actually said that if they give a booster, your antibody levels go up higher than even after that second shot.
That would be expected.
So, yeah.
Novavax is another whole interesting thing, because they're yet a third different vaccine technology.
Yeah, I think Novavax is super cool. It builds on a technology that we've had for a while. Our Robby virus is Novavax, vaccinated, which is very exciting. So we've got our own little human guinea pig. No data on how margaritas affect antibody levels, but worth investigating. I do want to raise the point. I'm going to actually skip forward a couple of slides. What do antibody levels mean? Because I think that's actually a really important question. So antibodies are these Y-shaped proteins that they are really good at sticking to things. And they're really good at sticking to things in a very choosy manner. So they stick to things like bacteria or virus or other infectious agents and tag them for destruction by the immune system. So in theory, more antibodies means more chances to tag it, more chances that it gets destroyed. But we know that that's not strictly true. So the best example that I know of this is people who have been vaccinated for measles. When you're first vaccinated for measles, you have a very high level of antibodies against measles. But as an adult, you have very few of them in your blood. There's not very many measles antibodies. But let's say your kid goes to daycare, somehow catches measles, maybe they're not vaccinated, comes back, brings it to you. Your immune system is like, holy shit, I've seen this before. And completely ramps up antibody production to fight off the measles. Your immunity lasts, even though you don't have the antibody levels that you would think gave you immunity. So we think of antibodies as what we call correlates of immunity or correlates of protection. That basically means that these two things often travel together, but they don't, A doesn't mean B. They just travel together a lot. So I
would also interrupt, sorry, but with regard to correlate of protection, like the people who study these things, like the physicians, the epidemiologists, like for other diseases, we have established, like the WHO has, this is the correlate of protection for Ebola. This is the correlate of protection for this. Like you could go into a population and say, what if there's a breakout in this town? And you can measure these levels. And there's scientists figured out what the threshold is. For COVID, that threshold isn't established. People have been trying to figure out, when are you immune to COVID? If they had this figured out, then my T cell project at my work would be canceled because there'd be no point. It's a new disease. We're figuring it out. In 5 years, we hopefully will be able to predict whether a person walking down the street would be fully resistant to COVID or just not there yet.
Brian, I just want to jump in with a slight correction. You're correct that we don't have the correlates of protection across the board, but the effort to calculate them or to determine them for COVID is massive. Some of the folks that I work with have actually analyzed over 30,000 samples of blood from people to do some of this work. And we actually do have correlates of protection for the Moderna vaccine. So they've been able to show a statistically valid way to predict whether a certain level of immune response equals protection against infection down the road. Pfizer, when they presented to the FDA last week, said that they have not been able to do that with their data. Wow. All the companies that have done phase 3 trials are going back and analyzing those samples again to see if they can do a statistical matchup. And it's really important because if we can figure out a correlate of immunity, that will accelerate the time to get new vaccines out or to get vaccines to new groups of people. You don't need to do a clinical study with 10,000 people. You could actually do it with a few hundred and have a pretty good sense of whether it works. And in an emergency, that's really important.
Yeah.
But there's a massive effort. And that's actually, again, your tax dollars at work.
Yeah. So there was a study published last week with Moderna showing correlates of protection between antibody levels and resistance to COVID infection in non-human primates, so monkeys.
Well, no, this has been shown in humans.
Yeah. So I haven't finished reading this paper because it just showed up on my to read list. But I've put a link for it if anybody wants to see it. I would say some of our antibody tests aren't like the antibody tests that they have in the UK is not very good actually. And that actually, I think that can be a problem because if you're saying like, oh, well, if you have 10,000 units of antibody, then you're immune. Well, how good are you at measuring 10,000? I think there's very good tests for labs like Agilent to do that with, but I think it's much harder in a point of care sort of environment. But I might be wrong because not that kind of doctor, unfortunately.
Testing is hard. Humans are different.
Yeah.
We could have predicted from the first thing was like, oh, there's this new disease and 1% of the people who get infected or less than 1% of the people who get infected die. And, a few percent get really sick and go into the ICU. And a big percentage of the people who get infected, they either have no symptoms or mild symptoms and they're walking around. It's like, if you just told me that about this disease, I'd be like, okay, thanks. The human immunology with regard to this infectious agent is highly variable. And we're still kind of battling with that. Like I got the Moderna shot. My son got the Pfizer shot. It is what it is. I felt nothing. He had a fever for 30 hours. And is he more immune than me? I don't know. We don't know. It was different. Even that was just the shot, not the vaccine. So,
yeah, my roommate got really upset because she's like, oh my God, I got the shot and I didn't get sick, so I'm not going to be immune. And I was like, hold on.
We don't know.
Like, actually, you're probably fine.
Yeah.
So I didn't see another thing in the chat. So I actually wanted to touch on the idea.
Yeah, we got to touch on mu.
On variants and get to mu and delta. So as you might have seen with COVID, a lot of people are now working on this. My roommate works in anthropology, and even her boss was like, oh, you should change your topic from like homeless students to like COVID students. And I was like, what? No, you're in like your 4th year. You're about to graduate. Please don't change your topic. But because a lot of people were working on this at the same time, people gave the variants different names. So 20I is alpha, which is also B117, which is also alpha, which is also VOC 2020-1201. These names, they're like puzzles. They do mean something to someone, and you can like tease that out. I don't think it's actually important. I am curious what the WHO is going to do when they run out of Greek letters, because they've already hit Zeta, so I'm not sure what their plan is.
They'll probably double them up. I'm just glad they got off the media was dubbing them, the British variant, the South Africa variant, whatever.
So,
and that's easy to understand, but that's actually part of the reason we've had so much trouble responding to infectious diseases in the past. Because you give it a geographic name, people think that if you're not going to the Ebola River, you're not at risk for Ebola. It's not that simple.
Yeah,
I agree.
I do think it's unfortunate because I think they probably should have gotten into that sooner.
Yes.
I suspect there was some aspect of like the UK being like, well, we don't want it called the UK variant. So now we have to come up with a new name for it, and it's alpha. Unfortunately, like class and socioeconomic status are also present in science. So how different are they? Oh, this is kind of grainy at the big size. So I think this is a little bit hard to read, but I like the idea. So it's the different variants are the columns, and then the rows are mutations that we know in the spike protein. So that's what's in the instructions are in the mRNA vaccine. So you can see, in some cases they share stuff like this T95I, is in iota and kappa. You know, I think the full figure is there. You can look at it later if it helps. I look at this kind of stuff, but it doesn't help most of the time. So this is
It is
analysis.
important, and just very quickly, because of all the testing that folks like you are doing, there's a whole team across the government that is looking at this data every single day, and you know, it's being factored into. I'm actually on my other screen. I've got different forecast models that take into account what variant is likely to emerge and how will that impact everything. So this kind of analysis is so critically important. I'm so glad that we're doing it, that folks like you are making a difference here.
Absolutely. And that's where, like, you know, now we've got like, so it's variants of concern, variants of interest, and variants being monitored. So that's kind of like different threat levels. Like, is it red, yellow, or green? So I just asked the question. I took the data set of there's about 3.8 million genomes of SARS CoV2 on this database called GISAID. It's the premier place to get this genomic information now. Government tax dollars actually from Germany, so thanks Germany. And I asked, well, how different is Delta compared to the original, or Mu compared to the original? So Delta in my calculations had 35 different mutations. Mu had 30. So how different are they from each other? So this is just the spike protein. So you can actually see there's 1, 2, 3, 4, 5, 6. Bottom of the screen is cut off, so I can't actually count it. 7 that are in Mu, but not in Delta. And I think it's 6 that are in Delta, but not in Mu. So Mu is now like the hot rage. This is what I get emails about at like 3 in the morning. Working with teams in Europe and China is a really great way to get panicked emails at all times of the night. So Mu in particular has these 2 variants. So this E484K. So I want to take a second. How do you read? Like, we see these changes in the news all the time, like, oh, E484K or N501Y. Like, what does that even mean? So the first letter is the amino acid in the reference. So E is
Glutamate.
Glutamate.
Too much detail.
Doesn't matter. 484 is the position. And then K is what it gets changed into. It means a lot of stuff to other scientists. I wouldn't worry about it, but that's what it means when you see those numbers, right? So Mu has these 2 ones in red, and those 2 ones in red have been shown to be better at escaping things like the monoclonal antibody treatments that we're using right now. Or they've been shown to be better at evading other immune system aspects. So that's why people are worried about Mu, because it has these things that in the lab we've shown that they make it better at escaping our immune system. I think it's important. So I wrote these slides today while I was in a meeting, so that's fine. So there's a study from Italy that they basically take blood from people who have gotten the vaccine, they mix it with virus, and then they mix that with cells. If the blood is capable of killing the virus or inactivating it, then very few cells die. If it's not very good at it, more cells die. So this is what's called a neutralization assay, because does the blood neutralize the virus? And in this study from Italy, they show that this lineage Mu still was, for people who had, I think this one was the Moderna, might have been the Pfizer. It was one of the MRNA vaccines. They were still able to robustly neutralize the Mu variant. So it might be better at escaping our immune system, but we're still totally killing it, right? Like, you know, instead of taking a really, really big hammer, you're still hitting it with a hammer. It's still fine. And I think that's actually really important to keep in mind, because people are really worried about Delta and people are really worried about Mu. And where is this going? And are we in for more trouble? So I think we can see some of this trouble potentially coming up, but I think it's also important to do these actual kind of practical tests that say it could be more of a problem, but it still works really well. And I think that's important to keep in mind. Any comments on neutralization assays from the immunology inclined?
Scientists do try to work on ways to model this in the laboratory and to predict better, to make better vaccines to keep this from happening. It's a lot of work. It's an imperfect process. That work's going on. It's not the same.
And protecting, we've got to do everything we can to reduce the number of cases that are transmitted, because every time SARS CoV-2 is transmitted from one person to another, every time that virus replicates is another opportunity for a variant to emerge. And on my other screen, I'm looking at a model right now that if a more dangerous variant emerges, comparable to what Delta has been compared to Alpha, we could be looking at 800,000 people in America dead in a couple of months. These are realistic projections based on the current vaccination rate and mask use and so on.
Yeah.
Absolutely.
The model I'm looking at right now is over 800,000 deaths by March.
Are those vaccines?
If there's no vaccines for children and a new variant.
So are you saying
a new dangerous variant?
Are you saying that a lot of those deaths are children, or are you saying like what is the makeup of that of 800,000?
So the issue with children is that if children aren't vaccinated and not masked, and the adults around them
they
are neither masked nor vaccinated, you've got a beautiful petri dish where the virus is spreading rampantly, which is what we've been seeing in the Southeast in particular. And we've got lots of new cases in those regions, which increases the odds of a new variant that is able to get around the immune response, either from the vaccine or from the body's own immune system. So it's kind of scary. We've got to stop the transmission.
Right.
I think that's actually really good. Let me find, I think it's one of the last slides that I put in. So talking about Delta versus like the original or versus Alpha. So viral loads, like how many virus do you have in your nose or in your lungs? It is around 1,000 times higher than the original virus. And, we consider that more dangerous, more infectious. Interestingly, you do see similar levels between vaccinated and unvaccinated cases. But in people who have been vaccinated, the virus levels go down much, much faster. It's sort of like you see a peak and down. Whereas people who've been unvaccinated, it's up and then it slowly goes down. So these are a couple of studies that fit that. This is one of the questions in the group. And I think it fit well with the idea of like, what does a more dangerous variant look like? Well, Delta is a more dangerous variant because there is a lot more virus, right? And the more virus there is, the more chance that you can catch it, transmit it, get infected with it, have a bigger response. So I think those are all good big questions.
So you're saying, just to repeat, Doug, the 800,000, that would be about 25,000.
That's cumulative.
So 25,000.
And that would be about.
I'm good.
It's cumulative, and that would be about 50,000 more than would likely happen without a new variant. And I'm actually looking at some other models now to see what some of the other variables are. So the University of Washington projections, which are a little different from the ensemble model that CDC uses, but their website's easier to use. They are estimating that if nothing changes, we're going to have about 780,000 deaths in the United States by the end of January. Sorry, by the beginning of January, which is just horrendous. We're currently at 670,000 or so. But if we were just to wear masks, we'd be able to bring that down by at least 30,000, just in a couple of months.
I know that there was a question that really talked about behavior, like the guidance on wearing masks. So
wear a mask, simple.
And no, but outside, period, outside, everywhere.
If you're
Sorry,
outside
I'll stop talking.
And you're not near other people, no, you don't need to wear a mask. If you're in a crowd, you're in a farmer's market or whatever, yeah, I would wear a mask if you're walking by people.
Okay.
Also, there was a recent study like a week or two ago that they looked at the different mask types. I know this was one of the questions. So N95s are the gold standard. That's the best mask probably you can wear. They're not very comfortable. KN95s are pretty close, not quite as good. The next best is the sort of paper blue and white surgical masks. And they're better than cloth masks, which is kind of surprising. You think cloth mask is kind of thick, but the surgical mask.
It's because they're multiple layers.
Exactly. They're multiple layers.
Surgical masks have 3 plies, and the nose piece is really important.
Yes.
You've got to tighten it over your nose.
Yep.
I mean, I would say I understand. I wear a mask when I go out. I get sometimes it's hard to breathe. My son and I, when we go on bike rides, we are those people riding bikes wearing masks. So I've done my own personal trial, and I can wear the surgical mask while walking, running, exercising because it has better flow than any of my cloth masks. And I was like, well, on the other hand, surgeons wear these for, 6, 8, 12, sometimes more hours. So it's a medical device. Like the surgical masks are made for being able to breathe easily for a long time while reducing risk of infection. Plus,
and really what we're trying to do is reduce risk. And there's a lot of different ways to reduce risk. And there's no one single way that will be a 100% solution. Vaccines will give you the most protection. Masks will give you a lot of protection. And depending on the type of mask, either a lot or not quite as much. Distancing gives you protection. Staying away from people who are exposed to COVID or otherwise risky. Those are all things. And what we talk about is the Swiss cheese approach, where you have layers of protection, knowing that some things are going to get through one layer or even 2 layers. But if you have multiple layers, something is going to protect you. So if you have a mask that only gives you 50% protection instead of 90% protection, well, that's still pretty good.
Yeah, it's not that hard to do.
Seems like the surgical masks are also the ones that are most readily available, right? It's almost like the no frills of masks, the surgical ones.
Yeah.
So
the manufacturing of them ramped up much faster. N95s are starting to be pretty well available. Actually, if you go to a pro-COVID area, you can get N95s pretty much anywhere. Because nobody buys them. I drove to rural Oregon and I got like a case of N95s at Home Depot. It was really easy. Back in Marin, sold out.
I have heard
I wish
good
I was
things.
kidding.
I have heard good things too about KN95, which is the equivalent,
Chinese equivalent.
Chinese equivalent, and you can get those really readily available off of Amazon right now. They ballooned in price for a while, but now they're back down to normal.
But even some of the N95s are back down in price.
Yeah, and you can if you're going to wear a cloth mask, I wear a cloth mask. You can get one with a pocket and put a filter in it. There aren't tests necessarily about that, I think, but like, again, it's like the Swiss cheese thing that we're talking about, right? Like if you're putting another layer between like you and COVID, it's only a good thing. I also included in the slide deck a test where they put different masks on people and made them run on a treadmill and measured their oxygen levels. And your oxygen just fine, even running on a treadmill with a mask on. I also put a link to the Swiss cheese model of COVID because I think the diagram makes it just super easy to understand. And I'll change this to edit. So then the other panelists can add things if they want, though, you know, that's always a little fraught.
No, it's okay. It's not, you know, being published. Yeah, I mean, I think the best mask is the one you wear. I think the best vaccine is the one you get in your arm. I think the best level of social distancing is the one that you feel is appropriate as you counterbalance in your mind, like, oh, I'm meeting with this person, or I'm visiting my grandma, or I never see these people, or everybody else in my party wanted to dine inside and I don't. Look, there's always risk. Nobody's perfect. We're all making trade-offs all the time. So I don't think it's just not binary, you know?
Have people heard about the micro-COVID project?
Oh yeah, that was kind of fun.
So the micro-COVID project, I think it's pretty cool. I know some of the people who are involved with it. So the idea is you enter your location, you enter the kind of activity that you're doing, your vaccination status, your mask, et cetera, and it makes a calculation of what your risk is for COVID. I think, you know, obviously it's not perfect. This is also a really like unfunded project, so they update as best they can. But I think it is a really good thing to get an idea to be like, oh, you know, I'm going to an outdoor concert. It doesn't require vaccination. Should I wear a mask? I mean, probably the answer is always yes, you should wear a mask. But if you wanted to quantitate how much you should wear a mask, micro-COVID, I think is really useful. Yes, thank you, Steve, for putting a link in the chat.
There's also a lot of variability depending on where you are.
So that's actually the first step, is that you put where you are and it looks up the prevalence in the area that you're in and then adjust for that. Because that's the thing, right? Like if you're in a hospital ward of COVID patients versus a Target in Marin.
Well, maybe not Marin, maybe like, I don't know, Santa Clara.
Yes.
Yeah. There's a lot of, and there's a big anti-vax movement in Marin, but anyway.
Yeah. Well,
you know,
yeah.
That's.
Well,
yeah.
But Marin is 90% vaxxed.
Right.
Yeah, which is kind of interesting. That's also sort of informative, right? Because you're like, well, what do I see in the news versus what does the majority of people actually do, right? Because in the news in the past, I was like, oh, you know, Marin School District has an outbreak of measles because kids weren't vaxxed, right?
Yeah, I want to comment on Eve. So Eve said they're in Maryland and the masking culture is very different from the Bay.
Yep.
So I've traveled recently. We've had some sickness in my family, so I've had to like fly out. And like when I was in Houston, I think I saw outside of the hospital, I saw maybe a handful of people wearing masks. You know, in the hospital, they mandate it. You know, same thing with like Oklahoma. I think even Portland, I saw a lot less people wearing masks than I saw,
surprising.
You know, in the Bay Area. Like if I go for a walk in my neighborhood, there's just, you know, it's very variable and like what you see in masks. It really heavily depends on where you are, whether people are masking or not. I'm going to continue masking for the foreseeable future. You know, even I think it'll be really cool to see this flu season as more people are back at work, but potentially masking. Do we see less cases of flu? Obviously, that's dependent on how bad this year's flu is as well. But, you know, plenty of countries in Eastern Asia have masked for many, many years and seen it be really effective as a public health measure.
Yeah, no, that's true.
Yeah, it was interesting. I was in New York City week and a half ago, and was in various neighborhoods. You know, New York City is pretty mask compliant, but the one neighborhood that like everybody was masked on the streets, in the stores, everybody 100% masking compliance, Chinatown.
Yeah, I saw this as well. I was in New York in March, and like Chinatown, even like street vendors selling new cheap NBA t-shirts were masked. But like I went to Brooklyn, and one of the like very posh neighborhoods there, and people were just like, "Oh, I'm vaccinated. I don't need a mask." And I was like, "Oh, okay, I'll wear mine."
Okay, so that's super helpful. So I think there was some more questions about Pam. You had a couple questions about you had a question about like how, which I think is important. I mean, I don't even know how to reconcile how people are moving through COVID as being, you know, like I can trust my body. My body's going to tell me. I mean, like whether or not I even want to get a vaccination. I mean, the misinformation and talking to people about this when it comes down to science, and even the videos that have been circulating are, there was some guy, Phil McSomething, that someone, you know, gave me hands, please watch this. As soon as he said Tucker Carlson, I was like, done. But how do we counteract what we're seeing around us, you know, right wing, left wing, the whole anti-vax sentiments? I mean, maybe that's really behaviorist more than it's a science question. But what do you think, Brian?
So I'm also interested in this exact same question, Bara. And, you know, I wish I had more anti-vax relatives or whatever that I could practice on.
Do you want to borrow mine?
But
Wow.
But it's like, but my brother's a sociologist, and I'm interested in psychology, and I'm really like, you know, with that big head off to Doug, I'm a scientist, but I think a lot about communication and changing the world, right? And it's like publishing some kind of data paper that gets published in this Nature journal is not really going to change the world outside of a couple of scientists, but communicating to real people does. So anyway, that's my preamble. I think, so I listened to a podcast called You Are Not As Smart As You Think. And they had a session on how to talk about vaccines to people who are vaccine hesitant. And the upshot is, don't go into a conversation thinking you're going to change anyone's mind. Put on like a growth mindset and say, I want to go into this conversation with the goal of creating a relationship with this person and understanding their viewpoint. And those are the only goals. And they give you some strategies. There's a specific strategy, I think it's called street epistemology. So, you know, philosophers in the audience. Anyway, there's what you do is you can ask someone like, oh, on a scale of 1 to 10, where 10 is, I want to be vaccinated right now, and 1 is, I'm never going to be vaccinated. How do you feel? And a lot of people would give an answer like 4. You know, it was like, and instead of asking them about their doubts, you're like, oh, well, 4, like, why aren't you 100% against vaccination? And you sort of, instead of doing my data versus your data, you get into this conversation about like, how do you trust information sources and how do you make those choices? And the conversation becomes not about the data, because whatever, we throw up the CDC and they throw up, I don't know what it is, Tucker Carlson or some YouTube or whatever. That doesn't matter. That's logos. That's other people's data. And if you can engage with people and be like, I want to understand you and how you make your choices, let's talk together And let's both self-examine how we make our choices of which data to trust. Because if you want it, there's data on the internet saying get vaxxed tomorrow, and there's data on the internet that say never get vaxxed, you're gonna die. So we all sort of have to make these choices. For me, I really hope that everybody gets vaxxed tomorrow, because my choices based on my data and my judgment system makes me feel safer. But I don't think I can converse usefully with the people who have other data who already rejected my data.
Babe, do you think you could find a link to that podcast and put it in the slides? That'd be great. Thank you.
Yep, will do.
I mean, that's one of the things that is really perplexing. And I mean, I'm working on an update of my book, which is a handbook for public information officers. And like in law enforcement and fire service, most of those folks never imagined they'd become public health communicators, but now they've had to. And on top of it, they're dealing with, in some cases you've got, a police spokesperson who's been asked to put out the messages for the public health department, but most of the people in their department are anti-vax or anti whatever the government says or anti whatever a democratic government says. And you've got people caught in the middle in ways that kind of just don't make sense. None of the plans that we've ever made for any kind of public health emergency anticipated this kind of divide where you have people who literally don't recognize the difference between facts and fiction. And you can't just say you're wrong, which is kind of where I automatically go, which is just, it doesn't work. I know it, but it's instinctive.
I mean, they tell you that too, right? The people
That's
on the
right.
other side,
We're
is
talking
the argument,
at each
right?
other.
But yeah, it's hard. It's a challenge. Okay.
Yeah. I thought it was actually really interesting. I think it was Wisconsin. They didn't have enough testing in their clinical labs. And so they trained some of their firefighters how to run COVID tests. And so these firefighters who, signed up for a lot are now being asked to do things that they never really anticipated before. I know a couple of the electrical vehicle companies in the US, some of them have big digital displays, they're partnering with the CDC and local governments to put up information about where can you get vaccines, what times, what is the safety information about vaccines. And, we're seeing all of these unlikely partnerships as people try and work together.
One of the other things which we've found, and the public health community has definitely been keying in on this, is that, yes, we have some percentage of people who are not going to get vaccinated because it's a political thing. And let's just put those aside for the moment, because I'm not sure I know how to change them. But then you have the other folks who just haven't gotten around to it yet. And that's a larger group than you might think, even today. So one of the things that I've been doing, and I hope others will do this too, every time I encounter somebody I don't know, I had a furniture delivery a couple of weeks ago, and I wanted to know if the folks that were delivering it were vaccinated, both because I wanted to assess my risk, but I also wanted to make sure they were vaccinated. Turns out they were not. So I made sure I had my N95 on and the windows open before they came in. But I also just said, okay, so, hey, how come you haven't gotten vaccinated yet? Would it help if I tell you where you can get the vaccine? And it turned out that they didn't have time for it. It wasn't a priority. They didn't feel like the virus was a threat to them. Yeah. So I shared some information about the Delta variant.
Furniture. They're busy doing their jobs.
Right. And if, they would have to take time off of work to get the vaccine. So I showed them how they could text their zip code to get vax, GETVAX, and don't ask me what the numbers are, or you just go to vaccines.gov, put in your zip code, and you will get 3 or 4 or 5 places in your neighborhood where you can walk in and get the vaccine. And that actually, and I actually got a text from one of the guys later that night saying, "Thank you, I'm going to get it."
Oh, that's so cute. I love it.
You're saving our lives. Good job.
I should have asked my movers.
I know.
But it's like we are at a point where we have to have one-on-one conversations like that, and it's like somebody posted in the chat with a model that has worked for some folks to be more empathetic toward the other folks. I have a hard time with that, so I'm not sure I can do that, but I can do this polite, one-on-one thing, and we really do need to go door to door. And I mean, the reason Marin's got to 90% vaccination is that they literally had folks, nurses and other healthcare professionals, knock on doors and talk to people one-on-one and answer questions.
Yeah, I absolutely think that's super important. I think, I think it's hard. I think, I mean, I wish it was more possible for more people to have the opportunity to go and get shots. I'm glad that shots are more freely available now than they were several months ago. It's interesting because California had the system where they were supposed to text you when you were eligible to get vaccinated. I never got a text for them to say that I was eligible, even when I was. But I do get a lot of messages from them to say we see, I used to live in East Palo Alto, which is a pretty relatively poor, relatively minority town, city. And they were like, can you go to your neighbor's door and just knock and be like, let me tell you about vaccines. Here's where you can get them. So it'll be interesting to see, right?
Yeah.
I think it's hard because we're pretty insulated.
And also, let's all admit that we're kind of tired of this. So kudos to Bara for organizing this, because it's tiresome. It's a lot. Everybody wants to be done with the pandemic. You know who most wants to be done with the pandemic? Whoever is the infectious disease chief physicians in all the ERs in all the world. They so want to be done with this, but we're not. And it's tedious, but we have to keep going.
Well, and this is something that I've seen too, is people are like, oh, the scientists told me that I didn't need to wear a mask, and then they told me I did need to wear a mask, and now they're saying I don't, now they're saying I do. And I think there is this concept that science is this endpoint kind of profession where you find the truth, and then the truth is known, and then you're done. No more. You retire, I was going to say buy a house, but not in the Bay Area. But science is constantly changing, right? Now we know that DNA forms this really cool spirally ladder thing, but some really smart people for a long time were like, no, that can't be how that is. It's definitely this other weird spiral thing. That's what the data says. Side
by side, like a zipper.
Yeah. And science.
It fits some of the data, but then the people supporting that, most of them died.
Yeah. Well,
so
I think
Helix won.
I think it's very important to recognize that science is an ever evolving field, right? We learn things as we go, or, maybe our tests weren't good enough before.
And the virus changes.
Yeah, well, that's exactly it. So actually, that's a really good segue to this piece of data that I wanted to show.
I want to show the graph. The graph is so awesome.
Let me see,
I
put
love
this
the
in
graph.
presentation mode.
Because I love graphs.
So this is from covariance.org. This is the percentage of cases that are different variants. So there's not a lot of data prior to January 2021, because as Doug said, we weren't doing a lot of good measuring at that point. But you see in April 2020, it's mostly this kind of Stanford red color. I don't know, a little more crimson, and that's beta. So, almost 75% of the cases were beta. And then, like out of nowhere, this green of Delta just destroyed everything. So I've seen a couple of different numbers. This graph, when you use the interactive mode, which I think is really cool, which is why you should go to covariant.org, it says that 100% of cases in the US right now are Delta. Based on my calculations, 98% of the cases are Delta. I think that's quibbling over 2%. But, it's very impressive. Like Brian and I were talking about this because when we were in undergraduate, admittedly at very different times, you'd see like, oh, well, evolution happens, but some new variant completely taking over the population is just unheard of, right? Like it'll be some, like the
it was
majority,
like a model, but it wasn't super common.
Yeah. Like you don't see it that often. And Delta has completely blown out of the water several really good evolutionary models of like how these things happen.
Have you mapped this against the transmission patterns? Because the geographical spread is really fascinating, and in fact, Delta overtaking was predicted very quickly once it emerged because we could see how fast it was spreading in other countries.
Yes, exactly. So, and it's really interesting because, so I don't have this figure, but I'll try and paste it in from GISAID, which is the place that collects all the genome data. They do a breakdown of regional distributions, and many places like Europe and North America, Delta is like nearly 100% of cases. But you look at places like South America, a third of the cases in South America are actually Gamma. Basically unheard of in the US and Europe, but that's where it originated, that's where it's spreading. And that's why, like with micro COVID, the first step is, where are you? I will post this figure in the thing right now.
One of the other really interesting things is that with retrospective testing, we didn't know about COVID early enough for lots of reasons, and I don't want to get into the China thing, but China published the genome on January 5th, and we didn't react until March 15th, basically.
Yeah, I started January, I think it was January 13th, based on my code database. But we have partners in China, right?
Mm-hmm. December 31st was the first United States media report of the outbreak in Wuhan. January 5th was when the Chinese scientists shared the genetic information that we were then able to turn into tests, should have been able to turn into tests. And anyway, but we've been able to go back and look at stored blood samples from random population groups. I know that the New York City Health Department did this, and we now know that COVID was here before February, and it absolutely came to the United States from multiple points, and primarily from Europe, not China. It got here from China too, but it was coming from Italy,
right?
Mostly Italy to the United States, Italy to New York and Boston, and then on to Colorado, before we were even aware of it spreading.
Yeah, absolutely. And there's also been really good samples with this, actually, with wastewater. So wastewater gets tested all the time for different diseases. So, once a month, every couple of weeks, people go in, get a sample of the wastewater, store it. And now people are doing tests on that wastewater. So you can reconstruct, like, how did COVID get to Italy? Well, it happened actually pretty early, and it happened several times. And they're able to sort of reconstruct this information.
There's some college campuses that are using wastewater testing to monitor
Yes.
for COVID as well.
University of
Yeah.
Arizona did group wastewater testing and was able to narrow it down to the specific dorm and then to the 2 people that were infected but asymptomatic, and have them quarantine, just stay in their dorm rooms.
They were able to do that from the wastewater sampling?
From
I thought
wastewater
the whole point
testing.
of the wastewater sampling was that it was pooled samples, so you couldn't trace it back.
Yeah, so they started with the pooled samples, and then they were like, well, we have the pooled samples from all of the dorms. What if we sample just the individual dorms? And then once they find the dorm, they test everybody in the dorm. And it was super cool to see
okay.
that.
So they went to antigen
They
or
went
other
in.
testing.
Yeah, they
Okay.
investigated it to be like, let's go.
Yeah.
Which I think is,
I was gonna say, because if they were gonna actually go into, like, each dorm room's toilet,
Yeah.
that's a little creepy.
no, this is like, they, after finding the data, they narrowed it down, which is super cool, right? And like, as a little point of pride, so the company that I work for is called Thermo Fisher Scientific. It is one of the largest scientific companies in the world. We actually provide training and materials and testing to universities at discount prices, and especially to historically Black colleges and universities. We set up a system where they get things for basically free, in addition to training and equipment, to do testing to allow students to come back to campus. Because, there are plenty of students that don't learn well over Zoom or learn well from a PDF. And I think it's been really amazing to watch big companies mobilize to make this possible.
I don't know if anyone I know that Amy just asked whether the likelihood of a monster variant. I know, Doug, that you mentioned the one that could lead to potentially 200,000 more deaths, but do we know what the likelihood of that actually is?
So just to be clear, what I was looking at was a statistical model. It wasn't looking at a specific possible variant. It was just that overall, the odds are there will be another variant, and a new variant will be more dangerous than Delta, which was more dangerous than the previous variants, and so on. And if another variant is about the same degree of badness to Delta as Delta was to its predecessor, that would lead to a lot more deaths if we continue to go around without masks and vaccines and so on.
All right, so I'm going to defer to Robby Virus on this because I don't have virus in my name. But so the monster variant, I think the monster variant that we're all hypothetically afraid of, why do I wear my mask when I ride my bike? It's not to demonstrate to my the people in Palo Alto that, hey, I'm pro-masks, like that's not doing anything. I wear my mask to protect myself because I'm a little bit afraid, right? But there's the whole virus infection, like what's the monster variant? Is the one that's going to infect you or the one that's going to kill you? All right, Rob.
Yeah, so the virus, this virus jumped from probably bats to humans. And so, it was adapted to bats, it was not adapted to humans. And so as the virus spread, it randomly tests out mutations, and some of these mutations gave it a survival advantage. And so, alpha and delta, were mutations that increased the ability of this virus to do what it wants to do, which is make more of itself. So how does it do that? It infects more people, makes people more contagious, and spreads to more and more people. That's what it wants to do. It just, the variant that can spread more is the more likely to spread. So viruses really don't want to kill you. If they kill you, you're kind of a dead end. If you're buried, you're not spreading new virus. They want you to walk around and talk to your friends and spread the virus to all of them. So the idea that, there's going to be a killer mutant that like is 90% fatal or whatever, it's probably not going to happen. Why, the virus doesn't care about that. The virus just wants to spread. So now a virus that, in order to spread, if it can overcome your immunity, it's going to spread better because the immune people now can start spreading. And, once you do that, but, the response of your immune system to the virus is so complex. Not only is it antibodies, but it's these things called T cells, which kill virally infected cells in your body. The idea that there's going to be some new variant that completely gets around the vaccines and, your immune system can't recognize, I don't think that's going to happen. I think, you might be more susceptible to it, but, the idea that there's going to be this super virus that is 50% fatal and the vaccines don't work, I don't think it's going to happen. And I don't know, maybe Anna has more to say about this, but I find it interesting, like Delta has completely taken over.
No flesh-eating diseases?
I'm sorry, what?
No flesh-eating vampires?
Well, that's another one.
Well, so
yeah, no
everyone-
flesh-eating vampire bacterial viruses.
That actually reminds me of a point from TWIV, This Week in Virology, great podcast, where for a while, their infectious disease expert was seeing a correlation between hair loss and people infected with COVID. So he said, wear your mask now or wear a wig later. And I think potentially if it made more people bald, we might see better vaccine results, like less vaccine hesitancy.
I don't want to, I'll die for the, but but I won't be bald. So I'm wondering, I've heard, just because of the migration and climate that there's just the possibility of like thousands of viruses that we haven't encountered yet, but there is one that's just gonna like totally kick our ass. So.
Oh my God, Brian, tell them about how much of a disaster bats are.
There are.
Rob's going to do that.
Literally
I'm answering a message.
Tens, hundreds of thousands of viruses out there that we don't know anything about. And they're in bats, they're in, God knows where. And the odds that another one is going to jump into people are pretty high, especially as we.
Very high. Yeah.
Because
also,
these animals interface with people more. It's, there's going to be a COVID-24 and a COVID-28. And, this isn't the last pandemic that we're going to face.
No, it's not even the last bat pandemic we're going to face, because bats are really weird. Like if you're into biology, bats have this really interesting, it's like they can slow down, they can hibernate, they can slow down their metabolism. They speed it up when they fly. They don't get a lot of cancer. Some small bats live more than 40 years. Bats are weird, but relevantly to pandemics, some bats live in colonies of millions. And then they fly around to other colonies. And that's kind of like people, right? That's like, if you're a virus, it's like, oh, this seems like a really good organismal system for me to infect. And bats have an immune system that's fairly tolerant of infection. We have this too, anellovirus. I bet if I took the blood of everybody here, I won't do this, but I could. If I took the blood of everybody here and I took it into my lab, and with some help, some computational help from Anna, I designed primers and I said, who's infected with anellovirus? Probably 90% of us have anellovirus, like DNA or RNA in our blood. It's like we have stuff in us all the time. Bats have a lot of that stuff, and bats have a lot of coronaviruses, and I don't know. Right? Yeah. Don't worry about it. It'll be fine. It'll be fine until the next time.
Well, there's actually some pretty cool evidence that like some of the viruses that we might have been infected with in terms like when we were back at like the era of the Neanderthals, which were not like Homo sapiens, but intermingled. And some of those viruses might have given us an edge, right? Because like the mRNA vaccine can't get into your DNA, but some viruses can. And they can sit there, they can linger, they can change stuff up. And there's some evidence that some of the viruses we encountered in the past made us more fit, which is really, really cool in a way. I don't think COVID's going to do that. So,
no, especially not now, right?
Oh my God, if there was a virus that gave you abs, think about it.
We should just come up with, you know, like the baldness thing. It's like, we've got to come up with what is it that's going to motivate people.
I know some people have
seriously.
actually, I know some people have tried this using the example of public health sectors convincing people to wear condoms, right? Like you don't shame people for not using condoms. You just provide the evidence to say like, here are the risks that you're presenting to yourself. And so the uptick in condom usage per generation is just huge. And then there was some pressure with this with COVID because like COVID may cause some infertility in men. Unclear how, unclear why. I think COVID is also one of those things like when you get millions and millions of people infected, if there's some weird side effect that could happen, probably happened. And we don't know how common it is, you know. I'd be curious to see.
Okay, there are some questions.
I'm checked. Oh, we got some questions. Okay.
Yeah, one of them is someone asked a little while up here about the graph that Brian loves that Anna presented.
Oh yeah,
If you could
that
put
Anna
the
said.
link
You can
to
find
that.
it. I love that graph. I love graphs.
And
Right.
Why vax people get infected, the antivirus response to us is, oh, okay, Brian, you wrote that. Okay. What other questions are there? So Steve is asking about this UW.
Yeah, Doug,
Yeah.
I think you got this.
Yeah.
Right?
Should we let Rob answer?
Yeah.
Rob, go ahead.
Rob, I want to touch on something that Anna said, because not all pandemics get the same press. So Rob, I'm going to give you a challenge. First, respond and then talk about HIV. Like why, like, could we have a pan coronavirus vaccine? And then what about he's a scientist. I'm giving him a challenge. What about a pan HIV vaccine? You can do this.
Coronavirus vaccine, I know there's been talk of this. I really don't know anything about it. I think it's going to be difficult.
I will say, well, why don't you list a reason why an HIV vaccine doesn't work particularly well, and then I'll respond with why coronavirus might work better.
Yeah, so HIV is a particularly, it's like the epitome of the virus that's hard to vaccinate against. Why is that? Because number one, it mutates incredibly fast. Like, you know, you see these coronavirus variants, HIV, you got variants, like a person that's infected like by HIV has their own variants forming inside of them. It just mutates like crazy. And so, you know, it's hard to make a vaccine because it just mutates like crazy, and, you know, it's going to get around the vaccines. And the other problem with HIV is it infects T cells, which are part of the immune system that fights viruses. So it's like, you know, HIV wipes out the cells whose job it is to wipe out HIV. And so, you know, you're just kind of fucked if you get HIV. That's the scientific term.
Yeah.
Well, but that's so you're correct about the vaccine there. And I actually worked with I was involved in the first phase 3 AIDS vaccine trial. which was very sad because it didn't work. But where we have done well with HIV is with the antiviral meds.
Yes.
Oh my God.
And I mean, HIV is a manageable disease now because we have these multiple drugs. And that's what we're going to need against COVID eventually.
Well, and that's the thing, like, isn't science cool that we were like, well, okay, we can't vaccinate against it because it changes too much, but we can gum it up. We can be like, oh, you liked that printer? Wouldn't be a pity if somebody put peanut butter in it. Bummer. Like, that's basically how some of the HIV antivirals work, right? Like, they're just like, oh, you can't use that. Too sad, unfortunate. But they do it in a way that's so much better than some of the chemotherapies. Because like the chemotherapies are like, oh, you grow fast, too bad, now you're dead. So that's why people lose their hair. That's why people become very sick. That's why they lose their taste. But the antivirals are very specific to the viruses, right?
Some of the cancer treatments that have radically changed cancer survival in the last 10, 15 years actually evolved out of HIV research.
Yeah,
because
which is just
we're
like
dealing with the immune system.
Well, and I think that's one
the
of
exciting thing for me is that the cocktail, the idea of multiple targeted therapies and having a range of choices that you can target precisely and change as you need to different targets or different therapies. That's the magic of science.
So I think that'll be something that's interesting to see in cancer, because now we're getting to this idea of, so it's called MRD, or minimal residual disease. So the idea is, let's say you have liver cancer. And so they say like, oh, your liver cancer should respond really well to this treatment. They give you that treatment. They check in 6 months later. How is it working? So the cells that are left over, that are still cancerous, that aren't responding well, that's the minimum residual disease. And so then they can look into that and say like, oh, well, this would respond really well to this other drug. So, I mean, sucks to be on 2 drugs, but sucks more to die of liver cancer. So like, I think it's really cool that now we're opening up this field. And this is the idea of, it's a buzz phrase that I don't like. It's democratizing sequencing. So the idea is making, knowing what type of liver cancer you have, it's kind of like knowing what kind of COVID you have. So you have to do these genetic tests. And once you can do those genetic tests, then you can target your treatment. We don't have that with COVID yet, but maybe in the future, right? You could say like, oh, you've got Zeta Tau. We need to give you not horse dewormer. Oh, let's see what Michael's question was.
Yeah, I think they do that for
Michael,
HIV.
do you want to just say what your question is?
Looking for it.
Yeah, I was wondering what the ultimate, what your opinion of the ultimate, what's going to happen? Is this going to eventually mutate to the point where it becomes something that the disease is not very serious, or are we going to get a universal antidote, or are we all going to eventually get it and get immunity that way, or what do you think the
I vote yes on all those,
I was just going to say,
except the
the-
antidote.
or vaccine, a universal
Right.
vaccine?
So there are, I think it's 4 coronaviruses that, predate, that infect humans and predate, COVID-19, SARS-CoV2. They cause common colds. And the interesting thing that I think is interesting, one of them clearly jumped from cows. It's related to cows. And if you sort of do the mutational analysis, you can date it back to like, it looks like it jumped to humans around 1890. In 1890, there was a pandemic that it previously thought was a flu pandemic, a respiratory disease. And it's like, maybe that was when this virus jumped from cows to people. It caused this pandemic. A lot of people died. Nowadays, it causes a cold. And I think the hope, one idea is, and the hope is that maybe someday SARS CoV2, which causes COVID-19, will just cause a cold because, you know, everybody will have had it and we'll all have immunity through vaccines or natural infections.
There's history of diseases becoming less. There's history of diseases becoming less serious. I mean, you know, what happened with the 1918 flu? Didn't that just sort of mutate into something less?
It did. There was a related flu.
Not until it killed 600,000 people, though.
Well, yes, but it didn't kill everybody, which is, I mean, that's sad, but that's the point.
Oh,
right? Like the virus. I mean, the virus doesn't want to kill everybody. It wants to infect everybody, and then sometimes our immune system.
The virus wants to live as long as possible.
Yeah.
If the virus killed you on day 2, you don't infect other people.
Right.
This is the problem we had with things like Ebola, right? Like Ebola was really good at killing people, and that turned out to be really good at not spreading as a result. Still terrible, but like could have been worse.
Could get worse.
Okay, so any last minute questions before our gratitude for this great conversation in year 2 of COVID? Any last minute questions? Do people feel are they satiated with new information, fresh from the source of these folks who are working so closely with the science and the journalism about it? And put it up if you've got a question. I just want to thank all of you, Rob, Brian, Anna, Doug, so much for the work you're doing professionally and for giving time tonight for sharing it. I mean, you guys have actually become like the rock stars along with the nurses and the frontline people. Like you guys are the rock stars of this age right now with the work you're doing. So thank you so much for the work and sharing it and making it so accessible, because it really makes it accessible to all of us. So thank you.
I want to tip my hat to the scientists, Rob, Brian, and Anna. It is so great to hear directly from people who are working on the things that you're working on. I mean, that's kind of what I do every day. So it's like real conversations with real people. We need more of this because that's how we will get more people to understand what's going on. And clear communication is awesome. And that's what you guys were doing.
Well, and absolutely thank you for I think there's several ways in which you make the information that we have more accessible, right? Like we're used to talking to other people who share our lingo. So like when my dad asked me if the mRNA vaccine would change your DNA, I was like, what do you mean? And he was like, I was like, oh, right. Okay. There are things that I know that you don't. Let me share. Real quick, I'm sharing the link again for us for the chat. I've added the contact information for myself and Brian. So if people have additional questions, these are our personal emails, not our work emails. So please don't sign us up for weird spam sites.
Let me ask you this. I can post everything that was in the chat. I can pull out all of the links, and I can just post it to the event. So if there are things that you don't want on there, one, you can make it so people can't edit it and edit things in, or you can actually everyone is on Facebook except
put our contact info in the chat. Yeah, Bara, if you could post that, that'd be great.
I'll be happy.
Because look, even if you do sign me up for some weirdo spam site, I mean, I have a Yahoo Mail. Like I've used the same email address for decades. I can handle it. Whatever, it's not a big deal.
There's also a really good Hank Green post.
I mean, I'm not Robby virus.
There's a really good Hank Green post, who's this amazing science educator, that he talks about there's this misconception that scientists don't want to share the results. No, get an interested scientist talking and they will not shut up. We're that. I don't know what to tell you. That's us.
Well, it's been really amazing, and I really appreciate everyone for attending. And I mean, we're all fighting the good fight, right? I would imagine that most of the people on here, the people that I know, are not just vaccinated, but they're pro-vax, most of them. And just, I really like, in some ways you're preaching to the choir, but it's really good to understand it better and to just be, you know, it has been going on a long time. And I think we are all tired, but the virus is not finished with us, right? It's not that it's not up to us. This is really, we're responding to the world we're living in. So I want to thank everyone. I mean, I can stay a little bit longer if people want to just schmooze, or if people have other things they want to talk about, but I want to just honor the time that our panelists have given us and be respectful of that.
Yeah, unfortunately, I should probably be up for meetings at 8 a.m. again. So, and Doug's at 5 a.m. So poor baby.
Yeah, well,
It's my routine. I've gotten used to it. Thank you for organizing this. Thank you, everybody, for the great questions. And Brian, Anna, Rob, I look forward to more conversations with you.
Yeah, absolutely. Same here.
Great. Okay, well, good night. Good night. Good night. Thank you.
Good night, everyone. Bye.
Thanks. Bye.
Cool stuff.
Bye, Rich. Good to see you.
Have a good night.
I was trying to get my friend Diane to speak up because she's a microbiologist, infectious disease specialist.
Here, I didn't see her
She
name.
was, yeah, and frontline provider because she's, yeah. 1 week she was pronouncing 2 people dead per shift. Now they're down at Kaiser. They're down to in Kaiser Sacramento, which is pretty small, one per day. So bad.
Let's stop the recording. Hold on. Just stop that. Let me see how to do that.